All right, I think we're ready to go. Welcome back. I hope everybody had a good break. The end is so near now. Okay. We left off partially through this chunk of material. So 13, 14, and 15 are the chapters. We thought about ways that we may combat some microbes with some physical means. We talked about in 14 some antimicrobials, how we might treat within our bodies. And it all ultimately is building up to the infectious disease, well, technically the immunology that we're going to hit next, and then some infectious disease. So chapter 15, the focus of this really is to think some details about how microbes have the potential to get in and take hold in the first place, all right? So we've looked at stuff, like I said, the physical means like disinfecting benches or different things that we may utilize to try to prevent or inhibit growth. But how is it that these things, these microbes have those potentials to take hold and really initiate the disease progression? When we wrap this up and transition into the immunology chunk of it, we will think about the mechanisms that our body employs to really try to combat these microbes and potentially to prevent this stuff from happening in the first place, from them being able to get in and take hold. So that's kind of the idea. And then we will wrap it up with kind of a quick overview of the plethora of disease-causing organisms in a last whirlwind of lectures. So, questions or concerns before I jump into this stuff? Okay. So, all sorts of interesting things. And as I was kind of scrolling through, just reviewing a little of this before starting this morning, there's a lot of stuff when you think about a clinical situation and you go into the doctor's office with whatever it might be that's ailing you. And there are lots of questions that are asked, and sometimes there are samples that are collected. And sometimes you may feel like there ought to be some samples collected, but there never was. But how is it that they actually know what the heck is going on in the first place, right? And some of this really is lots of schooling and understanding some of these mechanisms. There are definitely some overlaps with some culprits, some potential pathogens, where we may see some similarities in signs and symptoms, but some of the questions that maybe were being asked as we roll into the clinic help to guide them one way or the other as far as what truly could be causing some of these concerns. All right. So a lot of this stuff really, it makes me kind of curious. How do they know all of these things? All right. So lots of details where they're collecting information and some of the signs and symptoms. But like I said, we can see some overlaps with these things, and numerous things may be causing, like a cold, as an example, a plethora of different things that may be culprits for this. And now on our radar, is it COVID? Could it potentially be? Maybe, maybe not, right? And there are plenty of other things that bear lots of similarities with that, right? So further investigation potentially warranted to really get to the heart of what's happening, right? And it really depends on what it is we're thinking about. And like I said, maybe we have some specimens that were collected from these clinicians and maybe not. It depends on some of these circumstances. In this image, we've got two completely different types of conditions. One's infectious and the other not, all right? Both bad news for sure, all right? Our first player here is Plasmodium falciparum. This is one of a handful of culprits of malaria causing parasites. Plasmodium has numerous species that can cause malaria in us, and there are a handful of others that plague other animals as well, all right? And as far as malaria goes, falciparum, and we're not going to go into great details with this stuff right here, but falciparum is definitely the top of the list for the most dangerous, most severe forms of malaria. I feel like, you know, if you truly want to die from malaria, that's who you ought to get, right? There are some other malaria culprits that you feel bad, but not so much, all right? And the amount of red cells that they're getting in and infecting is much less than falciparum, all right? So here we can see one of the Plasmodia species. These guys want to be inside of red blood cells, all right? And at this point in this image, it's not, all right? We will talk some immunology stuff and then some disease as we progress through here in the next chunks of material. But oftentimes in these circumstances, when they're available in the circulation to be seen, if you will, by some of our own immune mechanisms, at this point, when they're not in the red blood cells, that's when you feel sick, when you feel the true ill effects. It's when they're able to be seen and targeted. And the fever and chill cycle that fits with malaria really is the result of these guys being visible to our immune response. And then shortly after they have caused those red blood cells to explode and be released out to be seen, they penetrate into new red blood cells and they kind of mask themselves. They're hidden away and your symptoms subside, maybe for a day, maybe for 2 days, right? Kind of depends on our species of malaria, all right? So there are potentials, like with these guys, to see numerous in a blood smear, numerous parasites, maybe free floating, or maybe inside of red blood cells. And in the case of falciparum, it's pretty likely that you would see lots of red blood cells with the parasites inside of them. All right. So all around here are red cells. All right. Some platelets. We don't see any white blood cells, but we do next door with the sickle cell. All right. You would probably expect maybe in the next field of view to start seeing some white blood cells. Typically in a blood smear, lots of red cells, erythrocytes, and then we start picking up the white blood cells, the nucleated cells, and neutrophils, as we see right here, some of the dominant players, and we'll touch on those in the next chunk of material, all right? So these, comparing them over here to the sickle cells, this, not infectious, this is a disorder where your erythrocytes, your red cells, aren't as flexible or pliable as they should be, and it causes them to have really kind of the sickle or sort of blade-like shape that's very impliable, very sharp, if you will, and it potentially causes the sickled cells to kind of clump up together. As we think about these moving into like capillary beds where they should be pliable and moving in there, they get stuck and they cause tissue damage with these blockages. So bad news, but not infectious, all right? And potentially looking at it through the scope, they bear a little bit of similarity, all right? Although you can see there's some details in here that you're not seeing over here, right? Nucleated and not, right? So same sort of morphologies, but culprits completely different, all right? So pretty interesting, I think. And really, you know, as we're looking at these things, there's a handful of different ways that we make some blood smears and stain them up that gives us slight variations in some of the color in these cells, but it's really the same idea. These all over here are the red blood cells. There's little platelets throughout. There's boatloads of platelets, and platelets are essential when we think about like blood clotting. These are fragments of bigger cells called megakaryocytes, and we'll talk about those guys briefly in our next chunk of material. But you would also, and we can see little bits over here as well, you'd expect to see lots of that stuff, right? So that's our first bit, looking at maybe some blood to try to evaluate things. And in a situation where we're thinking about some sort of infectious agent that's maybe in the blood, we talk about septicemia or being septic, and there are numerous things that could be the culprits for that. And some of them might be aerobic, others could be anaerobic. And so multiple blood cultures would be drawn from that patient and incubated at each respective situation, with air or without, and looked at, observed over the course of days to truly see if something's going on, all right? So truly depends on some of these circumstances. So this image is truly depends on the type of organism that we're looking at for some like of our time frame. We're not giving actual numbers here, all right? So time on the x-axis, and then we've got a couple of different things going up over here. So in red are the number of pathogen particles. These might be virions, it could be bacteria or fungi, whatever, all right? Or a little plasmodium parasites, right? Some sort of pathogen, all right? And then in blue, as we're looking at this, kind of mirroring our red figure, in blue is the symptoms that the patient is exhibiting. And they follow one another. That should sort of make sense. When we've got a boatload of pathogens, whatever they might be, one would expect that this is when you're probably feeling your worst, right? This is bad news, right? And then as the numbers of pathogens start to subside, we start feeling better as well, all right? This figure, if we were to kind of superimpose the bacterial growth curve that we've looked at before, it bears some similarity, all right? It's not exactly the same, but it's pretty close, all right? This incubatory period is kind of like the lag phase with those bacteria. When we first get whatever it is into us, it takes a bit of time for those pathogens to take hold and to actually start doing whatever it is they're going to be doing. So the incubation period depends truly on what it is, but maybe a couple of days, maybe we're talking weeks or longer for some of these incubatory periods, right? At this point, we don't feel bad at all, right? Symptoms, pretty much unrecognizable, all right? We just feel normal, all right? The number of particles starts to increase the littlest bit. We move into this next period, the prodromal, and this is when we start feeling maybe a bit under the weather, if you will, all right? As they're starting to replicate and ramp up their numbers, that's when we really start recognizing some of the symptoms in the first place, all right? We move into the real period of illness as these numbers have increased enough to actually start really making us feel ill. And likely at some point in here, might be when, if you're going to go to the clinic, when that starts to happen. All right. There are opportunities where maybe we go in and we see a negative test. We don't see the actual real culprit. And it might be because we're still early on in this infection and haven't had enough opportunity for the pathogen, whoever it is, to ramp up its numbers, maybe thinking immune responses, not enough time for that to truly take hold and start producing the respective players that might be needed for detection in the first place. All right, really depends on those circumstances. But as we move into this period of illness, greater likelihood that we might have some capabilities of actually detecting. And we will talk about different mechanisms in the next chunk of material, how we might utilize some of those techniques through immunology to evaluate some of that stuff. All right. So as we move up here to our peak, we think about, like in this growth phase, where those organisms start replicating exponentially or nearly exponentially. They're replicating very rapidly, and this is where we're moving up in this period of illness. Then you kind of start to plateau and hit that stationary phase. All right. At this point, there are numerous things that could be going on in our body trying to combat this. More immunology stuff as we think about our body's mechanisms of combating this, or maybe we've taken some antimicrobial agents and started to knock it down as well. All right. So we see the plateau up here, and this also kind of the peak of your symptoms. Right at the top of this one, there is the boatload of whatever pathogen particles we're talking about. All right. And then we see these starting to decline, and that's this next bit, which might also mirror that death phase of the bacteria if we're thinking of it in its little flask or whatever. All right. So period of decline, and then the convalescence. And as we move into the convalescence period, there are numerous things that still potentially we are capable of shedding, and we may be contagious at this point, or maybe not. It depends on the types of organisms as far as their capabilities of transmitting. But as we look at this, oftentimes in these windows are when it's truly more readily spread, but we could even move over into the prodromal. It depends on their respective players in this. When we think about stuff like the antibiotics that we talked about in the last chapter, oftentimes when we're trying to target these players, if we're thinking bacteria, it's on this uptick when these organisms are trying to pull in as many nutrients as they possibly can. They're replicating very rapidly. If we can drill them with some sort of antimicrobial agent at this point in this uptick, you've got a greater opportunity to truly target and get those drugs into those cells better than if we're on this period of decline, all right, where they're not metabolizing as well as they were over here. All right, so treatment truly depends on who we're talking about, but oftentimes thinking in the height of this would be the best time when we're thinking about utilization of antimicrobial agents. So I believe we talked about Koch's postulates early on. I think so. Is that true? Okay. I think this is interesting. And I like this image. I think it does a great job of showing what's truly happening as we move through here and think about these potentials. There are lots of infectious diseases that we think about utilizing Koch's postulates to truly get to the heart of what's going on. There are some that we don't, all right? But for the most part, when we think infectious disease, we utilize something like this. We talked before with like some of the mutations and maybe looking at chemical agents or something else like the Ames test, where we can see some salmonella and maybe making it revert back in these mutations if the chemical agent truly causes mutations, to show that chemical agents might be a potential problem, right? An agent of some sort of disease, but non-infectious, right? When we think infectious, Koch's postulates are what we employ. So here we've got two different situations. So we've got our healthy mouse up here, our healthy organism. We take some blood from it. Maybe we're thinking this pathogen's in the blood, all right? And we try to cultivate that. So we streak it out onto a petri dish, but nothing grows, all right? There are no pathogens in this healthy organism, all right? On the bottom one, we have the opposite. So we've got our diseased organism. We've taken a blood sample, and under the microscope, we make our observations, and up above, we didn't see anything, and down below, we do see something in the circulation of this diseased animal. So from that, we're able to culture it by making a smear onto this plate. Now in this just looks like an empty petri dish, but it's trying to just show there's no growth, right? So here we've got the organism isolated. There's growth on this. So the causative agent that caused this disease was able to be cultivated. Technically, there should be a image in here where we're going back to the microscope, right? And validating that what we saw here from this is what we're seeing growing on this plate. And then some more microscopy over here. Lots of microscopy truly should be happening as we're going through this, right? It's microbiology, right? So we would look at it and truly evaluate, are what we are seeing on this plate the same as what we saw from this diseased organism in the first place? We grow this stuff up and inoculate it, as we see over here, and we've given a different colored mouse to just show that, right? So we've inoculated this into a mouse, and it gets sick, and in this case, looks like it dies just like the other one, right? And we're able to cultivate from it the same stuff, all right? I assume we would take a blood sample from this mouse and we would look at that. So we would see this with red cells and green globs of whatever, right? Our microbes within there. And we would cultivate that blood and grow it up. And again, looking at it under the microscope. So a handful of steps in there where the microscope really should be in between, but that's okay, all right? As to simplify it, this is really what's happening, all right? See it, grow it, inoculate it, and regrow it. And it's the same stuff, right? And that in turn clues us into a boatload of infectious diseases that we've validated, that we're able to say this bacterium or this microbe, it doesn't have to just be bacteria, was what caused the disease. We were able to cultivate it and inoculate it and reobserve it, right? Showing that whatever it might be is a disease-causing agent. And I believe Robert Koch's initial stuff, I think it was anthrax that he was looking at, like cutaneous form of anthrax and able to transmit it. So we've recognized, maybe through Koch's postulates, that whatever it is a culprit for causing disease. But another question that we may want to address is how virulent or how dangerous might this pathogen be, right? Some things maybe we're more concerned about than others, right? As far as what sorts of impacts it might have on whatever we're talking about, probably ourselves, right? So The LD50 is the lethal dose. That's what LD is, all right? So the lethal dose, the amount of infectious particles, infectious agents that's needed to kill 50% of the population. So 100 lab rats or mice, and you inoculate a specific dosage in there. What is that dosage to kill half the colony? All right. That's what we're looking at with LD50s. All right. And there are lots of things that you can see, things that are potential problems where it may list off LD50s, all right, giving you some hints as some of the severity of whatever it is we're looking at, all right? So here we can see the number of agents, whatever it is, cells or virions that are needed, again, for 50% mortality in our experimental group. And in this case, 10 to the fourth cells or whoever, all right, are needed in order to see this level. And if we keep going out here to maybe the 10 to the seventh, we're approaching nearly 100%, all right? So you can increase it enough that it wipes them all out, but this is what we're looking at, all right? The LD50. So thinking disease, all right? There are, with our own bodies, lots of opportunities for pathogens to get in. And there's also lots of opportunities for them to get out. And we will see in a couple slides down the road, this same picture, but with maybe a few extra bits on there, all right? So these are the portals of entry, and we will wrap up today with the portals of exit, where we can see some similarities, all right? So there are lots of opportunities for organisms to get in, and our body has a lot of mechanisms in place to try to prevent this stuff from happening in the first place, all right? We will talk as we wrap this up and move into the next chunk of material back to this kind of idea, thinking about how is it that we may help to prevent things from taking hold in the first place, all right? Starting with the skin, and the unbroken skin provides a great barrier to the outside world, right? So broken skin, as we see here, needle sticks or insect bites, anything that could get something through this barrier provides an opportunity for that infectious agent to get in and take hold, all right? As we think about our face, the eyes, the nose, the mouth, mucous membranes are a great place for organisms to gain access to our body, and potentially some living there in the first place, right? Part of our natural defenses might be the microbes living on or in our body to help to keep us healthy in the first place. And when some of those get out of whack because of some of these pathogens trying to take hold, that may set the stage for a disease process as well. So we can look at other potentials. So in a female, the placenta can be a source for that unborn child. That fetus can become infected through some agents being able to cross the placenta, potentially, all right? And then anus, urethra, vagina, all potential portals of entry dependent upon whom we're talking about for being able to gain access. Lots of different players. And as we shift into the actual infectious disease, we'll start looking at all sorts of body sites and the potentials that some organisms may harbor. In this chapter, we'll talk about some of the mechanisms that allow them those opportunities to get in. And one of them can be a biofilm, a glycocalyx, a glue that holds them down and together and lends to like a colony of maybe multiple players participating in this biofilm and establishing true disease, right? Sometimes when we think about infectious disease, oftentimes, maybe like in labs and whatever, we're trying to evaluate one bacterium. We're trying to look at one thing, right? Your unknown is one bacteria that you're trying to figure out who is this, right? More than just one. Everybody's got some different stuff going on. It's not like everybody gets E. coli. That'd be super sweet though, wouldn't it? Like, man, mine's gram negative. So's mine, right? Mine fermented lactose. So did mine, right? That's not how it is, right? Probably multiple people in here have some E. coli as their unknowns, but there's other variations, right? So sometimes it might be just one culprit, but there are lots of opportunities when we think about a polymicrobial infection, numerous players, and trying to pinpoint the one guy isn't really sufficient because there's other players, other participants, and maybe the way that we go about fighting them would be different, right? So here we could see with this biofilm formation, some bacteria that are laying it down kind of looks smeared out on there. This is the sort of slime layer, the glycocalyx that's being laid down by these microbes, all right? And in this case, these are some biofilm that's forming in a catheter that lends to issues, right? With indwelling catheters, potentially lending to recurrent bladder infections, catheters that aren't something that's in place can have biofilms, but it's a little less than thinking about like an indwelling catheter, like going straight into the bladder. So I like this. I think this is super cool and disturbing at the same time. All right. Helicobacter pylori is a bacterium that lives in an area that we once thought you just don't live there, right? In your stomach is an environment that we would pretty much say that's inhospitable, right? Who is going to live in an area bathed in stomach acid, right? Really seems unlikely that anything could survive that, right? Can you survive that stuff? How could you, right? Some stuff, some pathogens, maybe some parasites that want to live in your intestines, need that stimulus of passing through the gut of something, touching that stomach acid. Very low pH might be what's needed to stimulate those eggs or cysts to start to open up. And as they make their way into the intestines, they're hatched out and ready to go, right? But Helicobacter doesn't do that. It lives in this environment and it's able to do it with some pretty brilliant mechanisms, I think, all right? So this we're looking at just a chunk of your stomach. So here's some epithelial cells that form your stomach. And it's lined with a boatload of mucus, all right, that helps to keep the stomach acid from coming in contact with these epithelial cells. It provides a barrier, all right? So this mucin gel is a very thick mucus layer that helps to keep our stomach functioning properly, all right? And Helicobacter, it wants to live there, all right? So it technically other stuff coming in contact with the stomach acid is going to annihilate them, but Helicobacter it's a little different, all right? So it makes contact with this mucin, all right? And it's should be impermeable to lots of things, but Helicobacter has an enzyme called urease. You guys did the urease tests. I think you did the urease tests before the break. And they were those little tubes that had sort of peach colored broth in there, sort of pea colored broth, right? And if the bacterium has urease, it turned it bright pink, right? And Proteus, I think, was who you guys had in the lab that should have turned it pink, right? And maybe it was E. coli. I don't remember who went in the other tube. You did 2 tubes. One of them was a positive and one was a negative, right? Helicobacter, we don't play with these suckers in the lab, but if we did, Helicobacter, when put into the urea broth, would turn it pink as well because of the urease activity, all right? Similar idea for how we might be able to identify those guys with that type of a test, right? So it's got this enzyme urease neutralizing the stomach acid, and it allows it the ability to get into the mucus layer to liquefy it because it's pretty impenetrable by itself. But with the urease and maybe some other stuff like mucinase that allows it to break these things down, numerous enzymes at play, allows it to bore in there and live, right? It also produces some buffering capacity that neutralizes the stomach acid so that it doesn't destroy it, and it allows it to replicate, all right? As it gets in there and whittles through this mucus layer, this provides an opportunity for stomach acid to make contact with these epithelial cells. Now we're talking ulcers, right? And this is painful, right? All sorts of potential issues, right? I think I mentioned this way back, maybe Koch postulate time weeks ago. Helicobacter pylori was identified by a couple of scientists that took some gastric secretions from a patient that had symptoms. They cultivated it, they grew it up in the lab, and they consumed it. They drank it down, and they got infected as well. They did Koch's postulates on themselves, all right? Kind of not a good idea, right? You shouldn't do science on yourself, right? You also really shouldn't self-diagnose yourself, right? WebMD provides so much stuff, but it also kind of makes us all hypochondriacs. We're like, good hell, I've got that, right? I know I do, right? There's issues with those things, right? But ultimately, recognizing that these bacteria are the culprits of this, and treatment with some antimicrobials, some acid blockers, I think drinking down a jug of Pepto, all of that helps to bring these suckers in check and get rid of these problems, right? Kind of crazy. So the portals of exit, like I said, similar to the portals of entry, and maybe a couple of extras in here, all right? So we've seen a handful of different things, this broken skin, needle sticks, these where we could be getting organisms in. We also could be releasing this stuff. If this pathogen is in your blood through a blood transfusion, it could be shared through shared needles or needle sticks. It could be transferred from one person to the next. In a wound situation, similar idea, that infectious agent, if it is there, could potentially be transferred to someone else. Insect bites, you know, malaria, as we started with this, is transmitted by an insect bite. And as it gets into us and replicates, when a new mosquito feeds upon us and sucks up some of these red cells, If it happens to suck up some of the plasmodia that's ready to replicate, then it does that in the gut of the mosquito and it becomes infected and is ready to inoculate that into somebody else. All right. So urethra, anus, the mouth, saliva, and sputum, secretions from the nose, or earwax, right? All some potentials. Tears, mammary glands, there are some potentials for infectious agents to be passed through the milk. There are other potentials through the placenta, vagina with secretions or blood, and back to the skin here. And this is just Blake's, so sloughed off skin. Maybe the potentials of stuff we could be thinking like some dermatophytes, some fungus, right? Athlete's foot or other potentials that may set the stage. So the ways that things get in, very similar to the ways things get out, not necessarily exactly the same. Potentially maybe some more mechanisms for things to leave. Truly kind of depends on these circumstances, right? There are lots of things, right? Thinking about inside of our body or within our blood. If you go to donate blood, they ask lots and lots of questions, right? About where you've been, what's happened, different practices, right? That maybe you participate within, just to see, is this stuff safe or not, right? I'm gonna end with one quick story. I donate blood. Years back, there was a little kid that was in need of a bone marrow transplant. And so there was a big blood drive here. They were collecting blood for Red Cross, but they were also doing some screening for bone marrow donors. And so if you were interested, you could have them take one extra vial of blood, and then they would do some testing to see if maybe there was a match. I think, unfortunately, they didn't find a match for this kid, right? But I donated for that and got on the bone marrow registry, and I get some messages from them, you know, just is stuff still current or whatever. You don't expect the bone marrow registry to do anything else, right? And when you make that first donation of blood for them to evaluate stuff, they pretty much told me, you'll probably never hear from us because it's just the way it is, right? And to find someone that's a match is pretty rare, all right? Close match, they try, but it is pretty rare, all right? So I left there with that in the back of my mind. And for years, I get, you know, messages and I'm like, yeah, it's all still current, right? And then one day, still back in the day with a landline and caller ID, all right? Hopefully you guys know that stuff, right? Maybe not, right? Our friends, their kid was calling his mom from the school office, and she couldn't hear a word he was saying or whatever because he had the receipt, he had it upside down, right? He didn't know what the hell. They'd never had a landline at their house. I'm like, what? You don't know how to use a phone, right? It's fine, right? So landline, right? The phone rings and I look over and on the caller ID, bone marrow registry, right? And my stomach kind of drops. They don't call you to see if your information is correct. They call you because they may want your bone marrow, right? I'm like, holy crap. I didn't expect that, right? The person on the other end goes through all of these questions. The first question is, are you still willing to donate? Because if you're not, she doesn't want to waste her breath, right? So then they go through all of these questions, travels, different, all sorts of things, right? Questions about lots of different diseases, right? So many questions, right? it was a very interesting conversation, and it was an interesting conversation for her as well, because as we were going through this and she's spouting off all sorts of different diseases. Have I had this? Have I had that? Whatever, whatever, whatever. And I'm answering, and I'm not asking what's this or what's that. There were a few things that I was throwing some comments back at, and she's like, wait a minute. I just have to ask, who are you, right? Who am I talking to? How do you know all of these things that I'm throwing out, right? All sorts of different blood pathogens. And I tell her I teach microbiology, and she's like, awesome. She's like, this is one of the easiest phone calls I have ever had, because I've got this whole list of trying to tell you what this disease is or isn't, whatever, and you know them, right? And I'm like, I think it's really cool, right? So I made the first cut, right? And she's like, just like I told you, you probably never get called. She tells me, you probably still aren't going to get called back. We will see, right? And as it turned out, the patient took a turn for the worse, and I was no longer on the radar, right? But crazy, all right? And all of that stuff kind of goes back to the very start of this, as we start thinking about going in and they start asking you all sorts of stuff. Some of it you're like, why the hell do you want to know that, right? Sometimes it's important, right? Truly is. All right, I will stop with that. We will wrap up the rest of this chapter on Wednesday. No class on Friday. The exam will open Friday. It'll be like the others, Friday to Monday. But if you're really on top of it, you want to take it Friday, you've got all the time. Good day. Peace be upon you. Enjoy the snow.